Cell cycle is very much essential for a particular cell for its division and produces new daughter cells. Cell cycle consists of two different phases. One is interphase, another one is M phase. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the cell cycle interphase. So let's jump into the topic. First thing, what is the definition of the cell cycle? A cell cycle is a series of events that take place in a cell as it grows and divides. So when a cell is going through a cell cycle, it actually grows and further it divides. And what is the time needs for cell cycle? It is extended for days, weeks, or longer depending on the cell type and condition. So if we take a look at the diagram of the cell cycle, so we have got a round shape diagram in which a little bit of space occupied by M phase. So this is the M phase, little bit of space, and most of the 90% of the case it is occupied by interphase. Okay, so this portion is interphase. Clear? So this portion is interphase. So interphase consists of 90% of the time of the cell cycle. Okay, so before I jump into the interphase, let's take a look some characteristics features of the interphase. So it is the period between two successive cell division. So it is a period between two successive cell divisions. Okay, chromosomes are decondensed and distributed throughout the nucleus. So in this phase, chromosomes are decondensed and distributed throughout the nucleus. Next point is, in this phase both DNA replication and cell growth occur in an orderly manner in preparation for cell division. So here major thing is DNA replication and also cell growth occur. Okay, next thing, cell grows in a steady state. So the nature of the cell grows is a steady state, and it is also consists of the 90% of the time of the cell. Interphase is subdivided into three subphases. Okay, so interphase is subdivided into three subphases, that is the G1, S and G2. Okay, so G1, S and G2. G1 is much longer than S or G2. Here is GS and next one is G2. So this is G1, this is S and this is G2. Okay, so this is S and this is G2, right? So three subphases are there. So G1 also stands for gap 1 or growth phase, or gap 1 growth phase or pre-synthesis phase. Okay, so pre-synthesis phase. It is known as pre-synthesis phase because in S phase DNA replication takes place here in the S phase. Okay, so the major characteristic feature of the S phase is the DNA replication process occurred in the S phase. So this is the synthesis phase, that is why it is known as pre-synthesis phase, and it is also known as gap 1 because it fills a gap between M phase and S phase. So mitosis, M stands for mitosis. So M stands for mitosis. So it fills a gap between mitosis and synthesis phase or DNA replication phase. So this one is known as gap 1. Another major thing is it is also known as growth phase. Why it is called as growth phase? Because in this phase the most essential molecules that is necessary for the particular DNA replication can be synthesized here, and those molecules are let me give you a chart of these molecules. Few of them are DNA polymerase. Okay, next one is the DNA primase. DNA gyrase, SSBs, that is the single stranded DNA binding proteins, DNA ligase, and many more. So the molecule which are actually necessary for the DNA replication, so those molecules are synthesis in G1 phase. That is why in this phase the cell actually grows and the volume of the cell grows and the cell is larger from the previous one. So this phase is known as growth phase, right? Next one is the replication phase. Here DNA replication takes place as sister chromatids are made, means one particular chromosome that replicates and produces its identical twins. Okay, so it produces its identical chromosome, and that is the identical copy. And these identical copies are known as sister chromatids. Okay, so these are the sister chromatids. So in here, DNA replication takes place, chromosomes are decondensed, and total DNA is replicated. Total DNA makes its own copy and produces a new chromatid or new chromosome that is attached with the same centromere, and those chromatids are known as sister chromatids as they are identical to each other. So this is the replication phase. Next one is the G2 phase. G2 phase is very much essential or very much necessary. That is, without the G2 phase, a cell cannot go into the M phase, that is a mitosis phase. So here G1 stands for growth and metabolism. So G1 stands for growth and metabolism. So here growth and metabolism takes place. S phase stands for DNA replication. DNA replication takes place. G2 stands for growth of structural elements. So here some structural elements are made up, and these structural elements help the cell to pass from G2 to M. Okay, G2 stands for gap 2. G2 stands for gap 2 or post synthesis phase. Okay, post synthesis phase, right? So gap 2 means it fills a gap between replication phase and mitosis phase. That is why it is stands for gap 2. And why it is post synthesis phase? Because the phase comes right after the synthesis phase. That is why it is known as post synthesis phase, right? So here some cells, so after the completion of the AM phase, actually two daughter cells are produced. Okay, so after the completion of the AM phase, two daughter cells are produced from the same cell, from the one cell, and some of the cells does not enter into the cell cycle, and they particularly enter into the G0 phase, that is the resting phase of a particular cell. So that is the resting phase for particular cells, like nerve cells, that is the neuron. Okay, like heart muscle. So heart muscle and nerve cell cannot go into the cell division. Okay, few cells does not have those G0 phase. That means they never enters into the resting phase. Those cells including skin cells. Okay, so skin cell divides 19 million skin cell per day. Okay, so 19 million skin cell actually produce per day, and those skin cells are rapid dividing cells and they cannot undergo those G0 transition. Okay, so here are three restriction points also present, or three checkpoints also present, that is totally observing or monitoring the cell cycle. Okay, so first one is the checkpoint is here, that is G1 is restriction point or checkpoint. So G1 is restriction point. In here, G1 is restriction point. Some proteins are activated like P53, that is the tumor suppressor gene, tumor suppressor protein. like PRB, retinoblastoma gene. So those genes are activated and they produce their particular proteins, and those proteins are continuously monitors the particular synthesizing molecules. That means the DNA polymerase, DNA primase, DNA gyrase, SSBs, DNA ligase, and many more. Helicases. So here is the helicase. Helicase actually built up by MCM proteins, mini chromosomal maintenance proteins. So those proteins are check the particular proteins that is produced in the G1. If all the proteins are matured and all the proteins have no deformities, then the restriction point can allow to pass from G1 to S. And here S to G2, S to G2, there is no restriction point present. But in case here G2M transition, here one restriction point is present. So this is the G2M restriction point. Here in G2M restriction point, this restriction point actually checks for some proteins, checks for some complexes, and if the complex is not much present in the cell, then the restriction point cannot allow the cell to enter the M phase. This particular proteins is known as MPF. Okay, MPF stands for maturation promoting factor. So maturation promoting factor is very much essential for the cell to pass from G2 to M by G2M restriction point. Okay, so maturation promoting factor are made up of cyclins and CDK, that is the cyclin dependent kinase. So maturation promoting factor or MPS promoting factor made up of cyclin and CDK. Cyclin is a protein and CDK is an enzyme. So CDK stands for cyclin dependent kinase. So it is actually a kinase that is totally dependent on cyclins. Okay, so this is the main thing of G2M transition. Another restriction point present in between M phase, this restriction point is known as spindle assembly checkpoint. So this is the known as spindle assembly checkpoint. So this is the spindle assembly checkpoint. In case of human cell, G1 consisting of 11 hours, the total cell division is 24 hours. Okay, G1 consisted of 11 hours, S consisted of 8 hours, and G2 consisted of 4 hours. Okay, and rest of the 1 hour is M phase. So M phase consisted of 1 hour. And here cyclins and CDKs plays a very important role in this transition, or G1 to S transition, or S to G2 transition, or G2 to M transition. So there are 4 different cyclins and CDKs present inside of the cell. Let's check about those cyclins and CDKs in a brief manner. So in case cyclin and CDK, okay, so in early G1, we observe cyclin D combined with CDK4 or CDK6. So in case here, when a cell particularly enters into the G1, then here in this case here the particular cyclin D and CDK4 or 6 combination make of, and particularly it helps the cell to proceed further. When it actually proceeds further in late G1, those cyclins D and CDK4 or 6 complex degraded, and then cyclin E and CDK2 complex is now formed. Here in late G1, cyclin E and CDK2 complex formed. The previous complex is also degraded and the new complex is make of, and it helps to pass the cell from G1 to S transition. Okay, and in next phase, in S phase, cyclin A and CDK2 found. That means when the cell pass from G1 to S, cyclin E degraded, and those CDK2 are actually now combined with cyclin A and make a complex that will help the cell to undergo S to G2. In case of G2, the cyclin is B and CDK is 1. Okay, so the cyclin A CDK2 complex now been degraded and cyclin B CDK1 complex is make up. Okay, and this complex help to cell to undergo the cell division process, that is the mitosis, and it produces cell until the next daughter cell proceeds into the next G1 of the cell cycle. Okay, so this is the total video of cell cycle. I hope this video is totally helpful for you to understand the various topics regarding the maturation promoting factor, cyclin, CDKs, the restriction points, and also the interface and the G1, AS, G2, all of them. If you like that video, please hit the like button. Don't forget to subscribe. Thanks for watching.